Okay, so get your notes. These are your notes. It says neural tissue, and we're going to start on them. So be sure you have your notes. It'll make it a lot easier to follow along. Uh, First slide here, the nervous system is a type of control center. It detects the changes of internal and external environments, interprets the change, and responds to the change. Now, if you look at the nervous system, you see that it can be broken down into the central nervous system and the peripheral nervous system. The central nervous system consists of the brain and the spinal cord. It receives all of the body's sensations. All nervous impulses to stimulate the muscles and glands comes from the central nervous system. The peripheral nervous system ties receptors and effectors to the central nervous system. Okay, receptors like, for pain, pressure, temperature, those are the receptors, and to the effectors like a muscle or a gland. Can be divided into the afferent system and the efferent system. The afferent system consists of receptors and their afferent or sensory neurons. So afferent neurons are the same as sensory neurons. The efferent system consists of efferent or motor neurons, which conduct impulses to muscles or glands. Now, one way I kept these two straight, if an efferent is the same as a motor and the afferent is the same as sensory, there's a word that will help you never forget which one is which, and it's the word same, S-A-M-E. Sensory is afferent, motor is efferent, so you'll never get them confused. The efferent system is divided into the somatic nervous system, soma means body, and the autonomic nervous system. So the somatic nervous system consists of motor neurons which conduct impulses to skeletal muscle. It's under voluntary control. There it is, controls voluntary movements. The autonomic nervous system consists of motor neurons that convey impulses to smooth muscle, cardiac muscle, and glands. It is normally under involuntary control. Now, the autonomic nervous system can be divided into two more, the sympathetic division and the parasympathetic division. The sympathetic division will gear you up for the fight or flight. Remember, fight or flight. It gears you up. Your heart picks up, your respiration picks up, and you're ready to duke it out. The parasympathetic division is rest or digest. The parasympathetic division is one that calms. It tends to calm the system down. Neuroglia. Neuroglia, the word means nerve glue, and it's oftentimes abbreviated as just glial cells. Function in the support and protection of the neurons. So let's look at the ones we have in our notes. The first one is the astrocyte. Astrocyte. It means star cell. For support, hold neurons close to blood vessels. And you can see that they're holding the neuron, which is yellow, close to capillaries here, which are purple. Maintain the capillaries of the blood-brain barrier. The blood-brain barrier prevents materials from getting to your nervous system like your brain. So it's very selective as to what can get to your nervous system. It's a capillary bed that is very selective as to what can leave, like medications. Certain medications have no effect on your central nervous system because they can't leave the capillaries. The blood-brain barrier won't let them pass. Astrocytes can make slight repairs to damaged neurons. Slight repairs. The next one, number 2, is oligodendrocytes. Oligo means few branch, and dendro means branches. So it's a few branched cell. Also for support, and they produce the myelin sheath. The myelin sheath of the central nervous system. The outer surface of the myelin sheath is called the neurolemma, and I think we'll look at that in just a minute. So the outer membrane of this cell that wraps itself around the axons and dendrites, the outer part is called the neurolemma, and the cell is a neurolemmacyte or an oligodendrocyte. Now we're going to see that there are gaps in between. So here are the wrappings here of these oligodendrocytes forming the myelin sheath, and there are little gaps in between. So it says the gaps in between the wrappings of oligodendrocytes are called neurofibril nodes. That's what they're called. I don't have it written down here, but neurofibril nodes, or the nodes of Ranvier. When the nerve fibers are myelinated, the region is called white matter due to the lipid content of the oligodendrocyte's membrane. If the region isn't myelinated, then it's called gray matter. And usually the cell bodies of neurons are not myelinated, and that's the gray matter. Number 3 is microglia. Microglia means small glue. These are phagocytic white cells. On this picture is picture B. They can freely migrate with the central nervous system. So you do have some white blood cells that can migrate through the central nervous system to make sure that any bacteria that possibly get in there are taken care of, or any damage that's happening in there can be cleaned up. Number 4, ependymal cells. Ependymal cells. Cuboidal to columnar cells which line the ventricles, those are spaces inside of the brain, and the central canal of the spinal cord. We will look at that on future slides. May be covered with cilia. You know, remember cilia is for movement, so that's going to require ATP, and those are microtubules that are embedded in the cell membrane that do require ATP for movement. Or microvilli. That's where the cell membrane is folded into little finger-like folds for secretion of cerebrospinal fluid. This depends on the location. So these ependymal cells can have cilia or microvilli, depending on where they're located. Now, the neuroglia of the peripheral nervous system is in your notes. A collection of neurons outside the central nervous system is called a ganglion. Now, ganglia are going to be collections of cell bodies of neurons which are outside of the central nervous system. And we will see some of those on future slides also. The neurons of these ganglia are covered with the wrappings of another two types of glial cells called satellite cells and neurolemmacytes, or Schwann cells. Schwann is an old name. Neurolemmacyte is the correct new name. These are the neurolemmacytes here, and their name kind of changes as to whether they're in the peripheral nervous system. They're called Schwann cells, and if they're in the central nervous system, they're called neurolemmacytes. So they have a special name, even though they are oligodendrocytes. Since oligodendrocytes produce a myelin sheath, well, there are two different areas that they do that in, the central nervous system and the peripheral nervous system. So the oligodendrocytes of the central nervous system are called neurolemmacytes. The oligodendrocytes of the peripheral nervous system are called Schwann cells, or just neurolemmacytes also. But they're the same thing. Demyelination. D means the removal, so the loss of the protective sheath, the central nervous system and the peripheral nervous system. This results in a loss of motor control and sensation. And some diseases can cause that: diphtheria, multiple sclerosis, Guillain-Barré syndrome. When the fibers are demyelinated, it's like this is shown in the picture. The impulses that are going along these fibers, they're electrical impulses, they can short out or be shorted out to the surrounding tissues or cross over to another neuron. And so the impulse is not directed to where it's supposed to be directed to. And the effect would be, loss of control of an area. Neurons. Neurons are nerve cells, the basic unit of the nervous system, and they conduct impulses. So we have three main parts of a neuron. The first one is called the cell body, the soma or perikaryon. Soma means body, so this is the cell body. Contains the nucleus, which you see here, that's a nucleus. Nucleolus, which is inside oftentimes. Lysosomes, mitochondria, Golgi, Nissl bodies, which are like ribosomes, and neurofibrils, which are microtubules in here. So this is the main portion that contains the nucleus. That's just a short way of saying that. You have Nissl bodies in your notes, like I told you, rough ER, rough endoplasmic reticulum, and free ribosomes, the sites of protein synthesis. Neurofibrils, the microtubules, are for support and transport of materials. So that's all in the cell body. Number two is a dendrite. Dendrite means branch. So you see there are dendrites that are sticking out on this multipolar neuron, which is what this one's called. These are branches. So branches from the cell body which conduct impulses toward the cell body of the neuron. So impulses are coming in toward the cell body from a receptor or from some other neuron. The third part is called the axon, A-X-O-N. A single long branch of the cell body which conducts an impulse away from the cell body to another neuron, a muscle, or a gland. So neurons have one axon. The axon hillock is where the axon is leaving the cell body, where it constricts as it leaves. That's called the axon hillock. It's the base where the axon leaves the cell body. Now, just like the muscle cells, remember they had some special names, like the sarcoplasm, and it was the cytoplasm of a muscle cell. Well, the cytoplasm inside of an axon is called the axoplasm. And the plasma membrane of an axon is called the axolemma. Axolemma. Now, these can measure from 0.4 inches to 3.28 feet long. So they can go from the sacrum all the way to your big toe. So they can be pretty long. The synaptic knobs, which they don't really show on this picture, but you know that they're at the ends of these telodendrites here, containing neurotransmitters, are at the ends of the telodendria. So these little extensions here are telodendrites, and they don't show on here, but you know from the previous chapters that at the end they're little bulbs called synaptic knobs. Synaptic vesicles are inside of these synaptic knobs. And remember they had acetylcholine in them. Collateral axons branch off the main axon. Now I don't have a picture that shows the collateral, but it would be, here's the axon, there would be another axon coming off. A branch off of the axon is called a collateral axon. And I do not have that on this picture. But you can look it up on in your book or on the internet. So let's look at the next slide here for nerve fiber. A nerve fiber is an axon and its sheaths. Okay, an axon and its sheaths. So here is an axon here. This model's been cut. This axon was cut off of over here. Here's the axon, and it has sheaths around it. And right now, it's just got the myelin sheath right there. the myelin sheath. So let's look at another slide to see this myelin sheath and how it's laid down, because this is a neurolemmacyte here. There's one neurolemmacyte there, and there's this nucleus always on the outside of the wrapping. Here's a second one here, a neurolemmacyte. It's wrapped itself around and around, and there's this nucleus always on the outside. And so the outside membrane is called the neurolemma. The cell is called a neurolemmacyte, and the multiple wrappings, that's called the myelin sheath. So on this slide here, the myelin sheath is a phospholipid multi-layer, many layers. It's been wrapped around and around and around, produced by the neurolemmacytes, wrapping around the axon many times. And this is showing like a program here. Here's a neurolemmacyte. See, they call it a Schwann cell, but that's an old name. Remember that. Neurolemmacyte is the newer name. And there's this nucleus there. You see it's being wrapped around, and it wraps around and around and around and around and around, and the nucleus is always on the outside of this wrapping, but you have multiple wraps there. It insulates and protects the axon, and it speeds the rate of impulse conduction. Now, because this is a myelin sheath, is multiple layers of cell membrane, it's got a lot of lipids there, and so this is called white matter. It tends to give the tissue of the nervous system a lighter color, and they call that white matter. Now on this page here, and like I said in your notes, the neurolemma aids in axon regeneration when damaged. As long as this neurolemmacyte is still alive, it can help to bridge the gap here and allow this axon to heal. If the neurolemmacyte is damaged and it's dead and this is cut, the axon will seal itself off and the impulses can't go from the left to the right because there's a break in there. So the neurolemmacyte can help to repair an axon if it gets damaged. It allows the axon more time to repair itself. Now the neurofibril node, or that node of Ranvier, that's an old term. The unmyelinated or fewer wrapped myelinated edges of the cell. It says areas between oligodendrocytes, between these neurolemmacytes. It's the little dips here in between. Those are called neurofibril nodes. They're wrapped, but not many times, and that's very important. Very important. We're going to look at something in just a little bit of why that makes the impulse conduction so much faster. Now we talk about axoplasmic flow and retrograde flow. I don't know what happened to my little deal here, but axoplasmic flow versus retrograde flow. So let's read the definitions. Axoplasmic flow. Okay, there it is on the top. The flow of nutrients down the axon to or toward the synaptic knobs. This is a one-way flow. So it just goes from the cell body down toward the axon terminals, the synaptic knobs. Now retrograde flow, this is the red line here, the flow of materials from the cell body down the axon, as well as materials flowing from the axon to the cell body. So it's a two-way flow. Materials can go down the axon toward the terminals here and the knobs, as well as from the terminals and the knobs back toward the cell body. So it's a two-way flow. And I've got it down here too. You see that the axoplasmic is the black line here. It's just one way, goes down to the terminals here. But the retrograde flow, the yellow lines, you see it's going from the cell body down to the terminals and back to the cell body. Now, classification of neurons. I have several definitions here. Now, the first one is this one on the far right, multipolar neuron. Multipolar, that means many poles, many extensions off of the cell body. A neuron with one axon and two or more dendrites extending from the cell body, found in the brain and spinal cord. This is the main one that we usually look at, is one like this, a multipolar neuron. Our model that we have is a multipolar neuron. The slides you looked at on tissues, that was a multipolar neuron. I couldn't tell you which one was the axon. I don't know who could, but only one of those extensions is an axon. All the rest are dendrites. That's why it's called multipolar. One axon and two or more dendrites. Well, your second one is a bipolar neuron. Bipolar. It means two extensions off of the cell body. A neuron with one axon and one dendrite extending from the cell body. Found in the retina of the eye, the inner ear, and the olfactory area. And we'll see those when we get to those senses. And then you have a unipolar neuron. So you have one extension off of the cell body. A neuron with one process extending from the cell body. The branch has two extensions. One acts as an axon, and the other acts as a dendrite. And you can see the dendrites moving toward the cell body, and the axon moving away from the cell body. Sensory and motor neurons. Remember, sensory or afferent neurons, right? S-A-M-E, same, sensory or afferent, transmit impulses from the receptors to the brain and spinal cord. Okay. This is not the best, but here's the receptors here to the central nervous system, brain and spinal cord. These are going to be unipolar neurons, so they're going to be sensory, just like this one right here, this unipolar neuron. These are going to be your sensory neurons here in this case. Somatic sensory neurons monitor the outside world and our position in it, and we'll talk more about those later. Visceral sensory neurons. What does visceral mean? Visceral means organs, and somatic means, soma means body. So visceral monitor internal conditions and the status of the organ systems. So we have several types of sensory. Number one is exteroceptors, exteroceptors. Provide information about the external environment, such as touch, temperature, pressure, sight, smell, hearing, and taste. That's the external environment. Number two is proprioceptors, P-R-O-P-R-I-O-C-E-P-T-O-R-S. Monitor the position and movement of skeletal muscle. Those are one type of proprioceptor. We have several we'll look at in this course. And then number three are interoceptors, I-N-T-E-R-O-C-E-P-T-O-R-S. Monitor the internal environment of the body. Digestive, respiratory, cardiovascular, urinary, and reproductive systems, as well as taste, pressure, and pain. So these are ones where you have discomfort internally, indigestion, cramps, that kind of stuff, or pain, that type of stuff. Now, motor or efferent neurons, motor or efferent, remember that's the part of S-A-M-E, motor efferent, transmit impulses from the sensory neurons to the motor, motor neurons, yeah. So they're going to transmit things out to a receptor, an effector, located in the brain and spinal cord. So they're going to be coming out. So number one is somatic motor neurons. Now, soma means body. So look at what it does. Sends impulses to skeletal muscles. This is under voluntary control. So somatic motor neurons go to skeletal muscles, and they're under voluntary control. But there are also some motor neurons that go elsewhere. Visceral motor neurons innervate all other effectors other than skeletal muscle. This is under involuntary control. So the visceral motor neurons are under involuntary control. Now over here, I have a good picture because we have, here's some receptors here. These are actually free nerve endings. You see the sensory, this is the dendrite of the sensory neuron, and there's the unipolar cell, and it's outside the central nervous system, so that's a ganglion. It's in the dorsal root ganglion. Here comes the axon into the central nervous system, and it's going to synapse with what's called an interneuron. Interneurons can be found between sensory and motor, so it's going to synapse between the sensory and the motor neurons, located entirely in the brain and spinal cord. So the interneurons are going to be in the central nervous system. They not only connect the sensory neurons to the motor neurons, but they can also send impulses or information up the spinal cord to the brain. So they have several branches. They don't just connect sensory to motor, but they have other branches that can go up and down the spinal cord, up to the brain or other muscles and down to other muscles. So they can divide that impulse. So here's your motor impulse coming out here, and that's a cell body inside the central nervous system. So here's the axon leaving and going out, in this case, to a muscle to cause it to contract. Now let's talk about electrical stimuli or electrical energy. Stimuli we receive is transformed into electrical energy. All types of stimulation from the environment. In your eye, light energy is converted into an electrical stimulus that's interpreted by your brain as sight, an image. The ear, sound waves that move through the air are conducted to an electrical stimulus. As your tympanic membrane vibrates from the sound making it vibrate, the auditory ossicles vibrate and send shock waves into your inner ear, which cause the cells in there to bounce, and that stimulates an impulse, generates an impulse. Movement. Electrical stimuli are generated to make muscles move. Okay, that's another type of electrical energy. Now I have dog whistle here. Most people don't have receptors for receiving that wavelength of sound. No electrical stimulus is generated, and so they don't hear it. Now there used to be a thought that elephants had some type of a connection, mentally connected, because these researchers would be on top of a mountain. They'd be watching two different groups of elephants, which may be a mile or so apart, and they would both start moving and go to the water hole at the same time. And they were trying to figure out whether it was a telemetry type of a connection. How did they do that? And they finally figured out that they emit low sound waves that are below our hearing range, but they can travel for quite a distance, for miles. And so they were hearing sound waves that we could not hear. And until somebody figured that out, it was a curiosity as to how they did that. So there are sound ranges that we cannot hear in, and other animals can. Now, if you look at this membrane here, in your notes it says the membrane, and it has a little line there, outside and inside. And then the reading here, it says there is a difference in the electrical charge on both sides of the membrane. This is called potential difference, or membrane potential. That's what the title of this slide is. Now, if you look at this one on the left, I wrote down here, potential difference or membrane potential. This is a resting membrane. The membrane is polarized, being more electropositive on the outside compared to the inside. So this is a resting membrane here. It's more electropositive on the outside than it is on the inside, and so it's polarized. By being more positive on the outside than the inside is positive, the inside is negative. If you look over here on the right, you can see kind of why. Just reading from your notes, sodium is high outside on the outer side of the membrane. So out on the outside, got a lot of sodium out there. Potassium is high on the inside surface of the membrane. A lot more potassium on the inside than the outside. This type of condition represents a form of potential energy. Now look at the definition here. Sodium ion concentration is high on the outside of the membrane, and potassium ion concentration is high on the inside of the membrane. These two gradients were set up by the sodium-potassium pump. So there is it's a membrane pathway set up in this membrane called the sodium-potassium pump. And we're not going to get real deep into that. We're just going to talk about the pump and show a very simplistic diagram of how it works, okay? But it's called the sodium-potassium pump. Now I have in your notes, sodium-potassium pump regulates the membrane potential. It pumps 3 sodium ions outside and 2 potassium ions inside every time it works. It works by way of active transport. That means it requires ATP, right? Active transport requires ATP. Always working to reestablish the membrane potential. High sodium on the outside compared to the inside, high potassium on the inside compared to the outside. So let's look at that. Let's look at that pump. We can see this sodium-potassium pump here. Again, this is just a representation, just a drawing to make the point. Now look at this. Here's the membrane. There's your phospholipid layer. That's the membrane. And here's the sodium-potassium pump they've drawn here as this little diagram structure here. And you can see that the cytoplasmic sodium binds to the sodium-potassium pump. All right, so starting off on the inside, the cytoplasm, sodium can bind. See how sodium's circular and it binds to this nice little circle here. So it picks up 3 sodiums. Next step. Sodium binding stimulates the phosphorylation by ATP. So ATP is broken down and phosphorylates the sodium-potassium pump. There's that little P there that's stuck to it. That's been phosphorylated, which means it's imparted energy to this pump to work. Phosphorylation causes the protein to change its conformation, change its shape, expelling the sodium ions to the outside. Now look at why they can't hang on. Look at the shape. It deformed the sodium attachment sites there, and they can't hold on anymore, so they have to be released to the outside. But look what happened to the potassium sites. Now they're in a big open V where they were a real tight V previously. So potassium ions can bind. So look down at this bottom right. Extracellular potassium ions bind to the protein, triggering the release of the phosphate group. So when they bind, that causes the phosphate to release, and loss of the phosphate restores the protein's original conformation. It pivots. And now look what happens. The sodium sites are now reformed and the potassium sites are deformed. Potassium is released and the sodium sites are receptive again, and the cycle repeats. And it just goes over and over and over. That's why I drew it in a little circle here. So that's just a real short little synopsis of how the sodium-potassium pump works. Every time it works, it pumps 3 sodiums to the outside and brings 2 potassiums to the inside to set up those 2 gradients, a high sodium on the outside of the membrane and a high potassium gradient on the inside. Now, let's look at this membrane a little closer. Here's the membrane here. I just drew a little sides here, but here's the membrane. The membranes have pores through which sodium can pass through to the inside of the cell. Here's all the sodium out here. I didn't draw any on the inside just to make a point. And there's a channel here. It's called a sodium channel. Calcium ions block the sodium channels. There's a calcium ion blocking the sodium channel, so sodium can't come. If the calcium ions are taken away, sodium ions can rush in by way of diffusion. Remember what that is? Diffusion, the movement of ions or molecules, in this case sodium ions, from an area of greater concentration, very high on the outside of the membrane, to lesser or lower concentration, practically 0 on my drawing. So they're going to go through that sodium channel if that calcium is taken away. When the membrane potential is established, there are more protons or cations, which would be sodium, on one side of the membrane than the other. Which side do you think has more positive ions, the outside or the inside? Well, because it's charged and there's more positive on the outside, it's going to be the outside. The membrane potential is measured in millivolts, and if we put a little volt ohm meter across this, you would see that we will be using minus 70 millivolts as our standard resting potential. So the resting membrane has a membrane potential of minus 70 millivolts. This is just like if you put a battery, meter across a battery to see what, if it was any good or not. You would see the needle deflected. Well, do you know what 9 volts is? If it goes to 9 volts, that's good, right? You put the battery tester on there. This is a small volt ohm meter here, just like you would use for testing your batteries, but it measures millivolts, thousandths of a volt. And electrons flow from negative to positive. That's why the needle is deflected over here to the minus side. They're moving from the negative side to the positive side. And a resting membrane, the potential difference between the outside and the inside is minus 70 millivolts. And that's what we're going to use. Having your notice positive and negative charges attract one another. This type of attraction causes a change in voltage on the two sides, and that's called current, right? Called current. Ion channels allow the passage of some ions across the membrane and control the passage of others. All right, we have passive channels or leak channels. Passive channels or leak channels are always open, but their permeability can be changed due to the proteins changing shape in response to local conditions. So they're open, but they could regulate the amount of, in this case, potassium and sodium ions going to each side of the membrane. They can regulate that. Then we have active channels or gated channels. They can be opened or closed in response to specific stimuli. I gave you, they exist in one of three states. Closed, but capable of opening. Okay, here's a closed here, but capable of opening. Or open, like the leak channels. So we're not going to look at number 3. Let's look at 3 classes of channels. I've got them down here in the bottom. Number 1 is the chemically regulated channels. Occur on the postsynaptic membranes. They open or close when they bind to specific receptor proteins such as acetylcholine, most abundant on dendrites and cell bodies of neurons. Number 2 is voltage-regulated channels. These are the ones we're going to deal with in this topic. Found on excitable membranes such as those of muscles and nerves. These membranes are capable of generating and conducting an action potential. And you see the channel can be open, sodium channel can be open and allow sodium to pass through, or closed. And we're going to see that that happens with the potassium channels also. They can be opened or closed. And the third one is mechanically regulated channels. Mechanically, when you talk about mechanics, you're talking about movement. It says open or close in response to physical distortion like pressure. So if you remember your lamellated corpuscles or the Pacinian corpuscles, those were going to respond to pressure. And even free nerve endings can respond to pressure for pain. Now we have a depolarized membrane here, and we're looking at graded potentials or changes in the transmembrane potential over a small area. Now we have, here is a resting membrane, which remember, calcium is blocking the sodium channels, and it's going to be minus 70 millivolts, and a depolarized, where the calcium's been taken away, that came back because at plus 30 millivolts, the calcium's come back. But let's look at the depolarized membrane. It's negative on the outside, positive on the inside. Well, electrons flow from negative to positive, and so there's your positive 30 millivolts. That's a depolarized membrane. So here we have a resting membrane right here. Let's just go through some of the things that are happening here. I think I can go through it right now. The resting membrane, let's just kind of rehash. It's minus 70 millivolts, you can see here. Oh, sorry. And you have sodium channels are blocked by calcium ions, and so the sodium cannot flow through the sodium channel. Here's your potassium channel, and it's deformed. That's why I have it crinkly there. It's all deformed. So right now, this is a resting membrane. No impulses are going down it. It's minus 70 millivolts. Well, a depolarized membrane occurs when remember the acetylcholine is going across the cleft on the previous chapter in muscles. When enough make it across, calcium ions start to pop off, and when the calciums are being removed from the sodium channels, sodium can leak in. And when sodium leaks in, the membrane is going to get depolarized. And there it is, plus 30 millivolts. That's the depolarized membrane. Well, at plus 30 millivolts, the calcium is going to pop back over the sodium channels, and look what happens to the potassium channel. At plus 30 millivolts, it forms, and now potassiums can leak out to reestablish the resting membrane potential. The outside is going to be flooded with positive ions to change the outside to be more electropositive than the inside. And there's your minus 70 millivolts. So the membrane has been reestablished. It's been repolarized back to the resting state. Now 18, this is slide 18. Things to realize. So number one is active transport. The sodium-potassium pump is working against a concentration gradient to reestablish the membrane potential. So it's going to require ATP. Remember, active transport requires ATP to move something against a concentration gradient from where it's in low levels to where it's in higher levels. And that's how the sodium-potassium pump works. If you go back to that slide, you saw that ATP was used. It is an active transport pump. Number two, diffusion is occurring. Well, sodium is going from an area of high concentration to an area of low concentration. That's when the sodium channels open. As a result, this results in transmission of the impulse. The membrane potential must be reestablished before any more impulses can be transmitted. So the calcium has to block the sodium channels again. The potassium channel has to open and let the potassium rush out to reestablish the resting membrane potential. So we have a little graph here. It's in your notes. And down here, I have it labeled resting membrane potential is minus 70 millivolts. And there it is. And then the threshold is minus 60 millivolts. So let's look at your notes here. A threshold must be reached before an impulse will be generated. This is called the all or none principle. Either the threshold is reached and an impulse is transferred, or it's not reached. To fire, in order for this impulse to be transferred, and for an impulse to happen, a threshold of minus 60 millivolts must be reached. That's the threshold. The required change of 10 millivolts is needed to generate an impulse and make the nerve fire. Now, if it's anything less than that, it goes to minus 69, 68, 67, 66. That's a subthreshold stimulus. You won't get anything. So when you look up here, let's look at your notes again. As a stimulus is applied, the membrane potential changes from minus 70, you see it's going minus 69, 68, 67. Where is the stimulus coming from? This is in your notes. I wrote this like a radio station. Think back, if you will, to last week when we discussed the myoneural junction. Remember the acetylcholine which diffused across the synaptic cleft to bind with this receptor sites on the postsynaptic surface membrane? You do? I'm so proud of you. Well, as acetylcholine makes it across the cleft and binds to the acetylcholine sites, the sodium channel proteins are affected and calcium begins to leave the channel. Now remember I said if enough acetylcholine makes it across. So this is what's happening here. Facilitation is happening. Since calcium is no longer blocking the sodium channel, it's open to the flow of sodium ions from the outside the membrane to the inside of the membrane. As a result, the membrane potential is changing from minus 70 toward the threshold stimulus of minus 60 millivolts. That's facilitation to the threshold. When minus 60 millivolts membrane potential is reached, the calcium ions in that area, they all pop off the sodium channels, and sodium rushes in, and the membrane potential depolarizes. And so when that happens, threshold is reached, all the calciums in that little area pop off the sodium channels, and sodium rushes in. It just floods in. And here sodium ions are rushing in. When it reaches the potential difference of plus 30 millivolts, the calcium ions block the sodium channels, so they pop back over the sodium channels. And the potassium channels are open now. Potassium ions are kicked out, so here they are, rushing out. And that's the downswing of this twitch. Kicked out to reestablish the resting membrane potential. And I just put roughly, this is just a rough number, 5 out, 5 in. That's just a general statement there. It is an impulse has been sent down the membrane. The sodium-potassium pump is consistently working or constantly working to reestablish the two gradients by pumping 3 sodium ions to the outside of the membrane and bringing 2 potassium ions to the inside of the membrane. If the membrane is poisoned, the nerve still works until an equilibrium is reached. And that's what some of these nasty nerve agents do, is they block the ability of the sodium-potassium pump to work and or block the sites so they can't open and close for letting sodium and potassium be transferred. And so you can't transfer impulses, which means you can't have muscle contractions or any movement. Well, here's another graph that I put on here, a little bit more descriptive. And what this shows, here's your resting membrane potential, minus 70 millivolts. There's your threshold, minus 60. And it goes along. Facilitation, this is at your bottom, but I defined it here. Facilitation, sodium ions slowly leaking in as more calcium ions, let's respell that, leave the sodium channels. This is facilitation to the threshold. So in my example, it reached the threshold. When the threshold of minus 60 millivolts is reached, as soon as minus 60 millivolts is reached, all the sodium channels open and sodium ions rush in. There it goes, up to plus 30 millivolts. At plus 30 millivolts, the sodium channels are once again blocked by calcium, and now the potassium channels open to reestablish the resting membrane potential of minus 70 millivolts. So the potassium channels open, potassium ions rush from the inside of the membrane to the outside to reestablish the resting membrane potential. But you can see that it goes down below and then it reestablishes itself. This is called hyperpolarization. Hyperpolarization is an overcorrection in the number of potassium ions kicked out. It is corrected by leak channels. So it corrects itself relatively quickly. So this graph fills in, by written information, what's happening during this muscle twitch. This is your impulse. The all or none principle. All or none, a threshold is either reached to cause a depolarization or nothing happens. It's like I want to stand at a light switch. I'm standing in front of a light switch and I think I want to turn it off. But I'm not really wanting to turn it off. So I'm maybe sending down some impulses, and some are making it across, but not enough to reach the threshold. But then I say, okay, I want to turn it off, and I send down a ton of impulses, and the whole area is flooded with acetylcholine, and all the calciums pop off, and sodium rushes in, impulses are generated, and my arm lifts up to turn the light switch off. So it either happens or it doesn't. You either reach the threshold of minus 60 millivolts, which would be facilitation to the threshold, or you don't. Facilitation toward the threshold. Didn't quite make it to minus 60 millivolts. So we have definitions. First one is threshold. Any stimulus strong enough to initiate an impulse. In our study, the membrane potential must be changed from minus 70 millivolts to minus 60 millivolts. The minus 60 millivolts is the threshold for our examples. Subthreshold, any stimulus too weak to reach the threshold, so it doesn't make it to minus 60 millivolts. Summation, when you add things together. you sum 1 plus 1 plus 1 is 3. That's a summation. So summation, many subthreshold stimuli, which will, when combined, reach the threshold. So you have several synaptic knobs releasing acetylcholine. In order to their combined forces will allow the threshold to be reached. Absolute refractory period, the time period when sodium ions are rushing to the inside of the membrane, and so the membrane is being depolarized. That's the absolute refractory period. That means that another impulse cannot do anything. The membrane is in the process of being depolarized, so another impulse won't make it depolarize again. Now, relative refractory period is the time right after the absolute refractory period. That's when it's starting to repolarize. You know, potassiums are flooding out. A stronger than normal stimulus is required to elicit an impulse. And if you remember Trepi, where you had one impulse, where it went like one time. Well, then you saw Trepi was when you stimulated it and it started to come down, it was stimulated again, and the impulse got stronger and stronger, and it'll eventually level off. Well, that's relative refractory period. That time period has been reached. Right after the absolute, it can be depolarized again. Now we have saltatory conduction. Saltatory means leaping. So saltatory conduction is the leaping of an impulse from one neurofibril node to another. This act greatly increases the speed of impulse conduction. So by being myelinated, you see this is myelinated, impulse conduction is rapidly increased. So here it is, neurofibril nodes. Remember, that's the space in between the myelin sheaths that are formed by each neurolemmacyte. There's a little thin wrapping area in between called the neurofibril node. Narrow gaps between the ends of the segments of myelin that insulate single nerve axons. And saltatory conduction is where the impulse can jump from one neurofibril node to another. Now that greatly increases the rate of conduction compared to depolarize, repolarize, depolarize, repolarize, depolarize, repolarize, depolarize, repolarize, depolarize, repolarize, all the way down this thing compared to depolarize, repolarize, depolarize, repolarize, depolarize, repolarize, depolarize, repolarize. It can jump quite a bit faster. And myelinated fibers allow impulses to travel very fast. So let's look at why. The speed of nerve conduction by fiber size. Okay, increased diameter of the axon increases speed because this reduces the resistance to the spread of charges by cable properties. Now, if you look at wires, large wires carry more current than small wires. And if you think about in the winter, we have a lot of what do they call them? Room heaters, space heaters that catch houses on fire. Now, why do they do that? Because someone puts an extension cord, like a lamp cord, between the wall and plugs that space heater into that extension cord, which is a small wire. Now, the wires in your wall are larger, and they can carry more current. So that space heater is trying to pull more electrons through that small wire, and the wire gets hot. If you increase that to a larger wire, those are like the orange cables, you won't have that happening. So let's look at that. The diameter of a fiber affects the speed of conduction. So let's look at the first one I've got here, an A fiber. That's what goes in your blank, A fiber. Large fibers, now they've got 13 to 20 on here, that's fine. All are myelinated. And look at this, read the speed of conduction. Now they've got 80 to 120 meters per second. And as you're going to find, those are ballpark numbers. My notes have 140 meters per second. That's fine. I'm not going to be specific on a test as far as the meters go. But that's like more than a football field per second. And that's in your body. So those go pretty fast. Those are very large. Found in large sensory nerves for like touch, pressure, heat, and cold. And in all skeletal motor nerves. They connect the brain and spinal cord with sensors. Okay. And let's look at B fibers. You can tell smaller diameter than A. Middle diameter fibers. Now they have 2 to 6. I've got 2 to 4. That's okay. And they've got 18 meters, and here they have 35 to 75. That's fine. That's fine. It's just to get the point across. Transmit impulses from the skin and viscera organs to the brain and spinal cord. So those are a little bit slower, but still 35 to 75 meters per second. You know, we're not even 2 meters tall. So yeah, that's pretty fast. And you go to C fibers, small diameter fibers. This one is myelinated here, but my notes say unmyelinated, 1 meter per second. Now this one's a small one, and it has 1 to 5. So this is a small fiber or even unmyelinated. They're very slow. Are involved in constricting and dilating the pupils, changing heart rate, and urinary bladder. So those are some areas you could think about that have the small fibers. Excitatory transmitter-receptor interaction. Okay, you got to break this down. Excitatory transmitters. Well, we've talked about acetylcholine. Acetylcholine is an excitatory transmitter, and how they interact with the receptors. So that's what this phrase means, excitatory transmitter-receptor interaction, how the transmitter affects the receptor. The ability of the transmitter from the presynaptic membrane, that'd be like the synaptic knob, to raise the postsynaptic membrane potential enough to reach a threshold. This requires a strong enough impulse to release enough neurotransmitter substance. So let's look at this. Oh, that's what I was right there. Okay. Your first blank is facilitation, and I've got it in bold print right there. The act of raising the membrane potential close to the threshold level. That's going to be facilitation. I have one condition is raising the membrane potential toward the threshold but not reaching the threshold. This is called facilitation toward the threshold. So if it does not reach the threshold, it's facilitation toward the threshold. If it goes to the threshold, that's facilitation to the threshold. That's what I put down here too. So we have some terms. I have the combined effect of several synaptic knobs acting at the same time to raise the membrane potential to the threshold and initiate an impulse is called summation. Summation, the additive effect. One form of summation is called spatial summation. That's your next blank. This is the accumulation of neurotransmitter substance from several synaptic knobs. Spatial summation, the additive effect. Temporal summation is the accumulation of neurotransmitter substance from a single rapidly firing synaptic knob. So it's really trying to release a lot of acetylcholine to cross that cleft, but it's working by itself. The next blank is synaptic delay, which is what this is a definition of. The time required for the impulse to cross the synaptic cleft is called the synaptic delay. The impulse crossing is when the synaptic knob releases acetylcholine into the synaptic cleft, and it diffuses across and binds with the receptor on the postsynaptic membrane and generates an impulse. That's called the synaptic delay. Now, I gave you some excitatory transmitters. Adrenaline and caffeine cause facilitation by competing for acetylcholine receptor sites. They mimic acetylcholine. I used to work with some guys when I first started working for the college, and they would drink several of those aluminum coffee carafes, those big old aluminum cylinders of coffee a day. So they had a tremendous amount of caffeine in their system, as well as they were allowed to smoke in the building at the same time too, so they had nicotine. And one in particular smoked cigars, and he drank an awful lot of coffee. He had so much caffeine in his system that he shook. And you could not ask him to write something down because it was like chicken scratch. I mean, you couldn't read it. Just a whole bunch of up and down jaggedy marks. And so we would just tell him, just tell me what you want me to do. Don't write it down. So he had a lot of caffeine, which was sending false impulses across that synaptic cleft. It was there in the cleft, and it was triggering false impulses to be generated. Now, calcium is removed from a nursing mother's body which causes false impulses, which is cramping. Remember what blocks the sodium channels? Calcium. So you start removing too much calcium, you get false muscle contractions. Inhibitory transmitter-receptor interaction. So there are inhibitory transmitters also. A reaction which makes the resting membrane potential more negative and therefore making it more difficult to reach the threshold level. If the change was from -70 to -90 resting membrane potential, it will now require 30 millivolts of facilitation to reach the -60 millivolt threshold, whereas previously it only required 10. So it's harder to generate an impulse. Now, muscle relaxants and depressants cause a change in the resting membrane potential to levels less than the resting -70 millivolts. Now, if the change, like it's telling you here, was from -70 millivolts to -90, now it's going to require 30 millivolts facilitation to reach the threshold, which is what this statement is saying right here. It just makes it harder to move. Okay, that's what it makes. That's what muscle relaxants are supposed to do, make it to where you don't want to move. Integration at synapses. These are some examples here. A postsynaptic neuron is affected by excitatory and inhibitory substances from the presynaptic neurons. Okay, so there are inhibitories that can be released also. This neuron that I've got right here, it's my little circle here, represents a neuron, requires 5 excitatory neurotransmitter substances from the presynaptic neurons. So 5 of them have to be firing, okay, in order to reach the threshold. It has to have a +5 excitatory neurotransmitters. So I defined them here. Excitatory neurotransmitters are the blue squares. Inhibitory neurotransmitters are the red squares. So let's look at our if statements. If the excitatory, which are blues, which there's 7 of them here, is greater than inhibitory, which are 3, and they are, but less than the threshold, let's see, each excitatory and inhibitory cancel each other out. So you cancel 1, 2, 3. Oh, so you got 4 excitatory. That's less than the threshold. That's facilitation. Example 2. If excitatory is greater than inhibitory, okay, look over here, there's 7 excitatory, that's greater than 2 inhibitory, and greater than or equal to the threshold, then the impulse is transferred. Okay, let's see. Take 2 of these positives off. How many excitatories do we have left? 1, 2, 3, 4, 5. That's what it took. It was equal to the threshold, so the impulse transferred. If the excitatory is less than inhibitory, excitatory is 4, inhibitory there's 6. So you have 2 inhibitories right here, greater. So the net effect is 2 inhibitories. The impulse is not transferred at all. So it canceled out all these excitatories, and there was 2 inhibitories more. So that's going to be like a negative number. So impulse is definitely not transferred. Some transmitter substances. I just wrote some down here for you. Excitatory neurotransmitters. And there I have acetylcholine and norepinephrine and epinephrine. You see, acetylcholine can have an inhibitory effect on the heart. So don't do that. Inhibitory neurotransmitters. Here's one called GABA. Gamma aminobutyric acid. That's why they abbreviate that, GABA. Glycine and serotonin. Those are some transmitter substances. Now here are transmitter substances and neuromodulators. So I put both of the transmitters on here, excitatory and inhibitory, but a new one called a neuromodulator changes the nerve somehow. Compounds which influence either the release of the neurotransmitter by the presynaptic neuron or the postsynaptic cell's response to the neurotransmitter. Okay, let's look at endorphins. That's your blank, endorphins. Are like enkephalins, beta-endorphin, and dynorphin are produced by the brain and the spinal cord. We have a holiday coming up called Thanksgiving, and you probably noticed that when you eat a lot at Thanksgiving, Everybody's going to try to go watch a football game or something. They end up falling asleep on the couch. Well, endorphins are released as a result of that meal that you ate. Endorphins are released. They relax the body. Here's what they also do. They inhibit the release of substance P at the synapses that release pain. So endorphins are going to be molecules that reduce pain, as well as they relax and reduce pain. Now, if you remember from general biology, we had endorphins there, and there was a molecule which mimicked endorphins. And the molecule which mimicked an endorphin is a synthetic that was produced called morphine. Now, they have a site, endorphins and morphine, have a site that binds to the presynaptic membrane to prevent the release of substance P. So morphine mimics endorphins. When I broke my elbow, they put me on morphine because I wasn't producing enough endorphins to block that pain. That's substance P. And so the morphine did, and I had very little pain from the surgery to repair my elbow. Now we have something that's before here called regeneration. This process can occur only if the cell body is not damaged, cell body of the neuron, and if the oligodendrocyte does not degenerate, if it hasn't been damaged also. The oligodendrocyte can proliferate, can repair itself, and form a solid core around the axon, allowing the axon time to repair its axolemma, its membrane. If the oligodendrocyte is damaged and it dies, then the axon can't repair itself and it seals off both ends where it was cut in two, and then impulses are not transferred down that axon anymore to the effector. We have neuronal pools. These are organized collections of neurons. Now we have two definitions. A discharge zone. This is an area that has enough presynaptic knobs present to generate an impulse on the postsynaptic membrane. So this is where an impulse is transferred. It's called a discharge zone. If an impulse is not transferred across that synaptic cleft, it's called a facilitated zone. An area that has enough presynaptic knobs to raise the membrane potential, but not enough to generate an impulse, not enough to reach the threshold of minus 60 millivolts. So let's look at some of these neuronal pools. The first one is a simple series circuit. And this is C here on your pictures here. They call it serial processing. A presynaptic neuron stimulates a single postsynaptic neuron. There's the presynaptic neuron, the postsynaptic neuron. There's your synapse in between. A divergent circuit. Look at picture A over here, example A. One neuron stimulates two or more along a circuit. So that's stimulating two on this example here. Number 3 is a convergent circuit. Now we have convergent circuit right here where it says parallel processing, which I've also got. It's a type of conversion, but I'll look at letter B, convergence. Impulses are received from the same source or several different sources. So several different sources are sending an impulse to one neuron. They're converging. The impulse is converging on one. A reverberating circuit, a positive feedback circuit. The initial impulse keeps perpetuating the cycle that forms a loop. So here's neuron A sending an impulse down to neuron B. Now neuron B is sending it to C as well as back up to A, which sends it back to B, and B sends it down to C as well as back up to A. So that's a reverberating circuit. It's a loop. The parallel after discharge, here's a whole big picture here, the parallel one. This is divergent for one thing. Parallel after discharge circuit, one neuron, and we've got two here, sends an impulse to several other neurons, and there they are, which in turn convey the impulse to a common source. And these don't show the common source, but they would be going down to a common source. So a parallel processing would be kind of like a divergent and then a convergent together. That's parallel after discharge. Okay, so that's the end of the neural tissue notes. I'll put the other ones up in just a minute.